Hello and a very warm welcome to our webinar today. We really appreciate you joining for our topic today of multi-organ chip for improving drug safety during pregnancy. Just for the start, a bit of housekeeping. So you will have noticed there's a control panel. In there you can see a chat function. So please use that if you've got any requests during the webinar, and I will try to get back to them really quickly. And there's also a Q&A function, so we'd appreciate if you use those for any questions that you have to our speakers. And once they're finished, we will do a Q&A session, and we will go through all of them as time allows. And yeah, lastly, we are recording all of this, and I will send the recording to you after this. So if you want to share it with your colleagues or revisit it afterwards, you will definitely get that. So before I introduce our speakers, we would like to find out a bit more about you. So we've prepared actually 2 polls. And the first one would be, we're interested to learn which industry you're working in. So are you from academia or in industry like pharma or CRO? I'm really curious to find out about this. There's a lot from academia here already. Very good. Some from pharma and biotech as well. Perfect. Okay, so while the results are coming in, I might actually start with introducing our speakers already. So first up, we have Katja Graf. She pursued her studies in biochemistry at the Friedrich-Schiller University in Jena, where she also earned her PhD from the Leibniz Institute for Natural Product Research and Infection Biology. Her postdoctoral research introduced her to the field of organ on chip, which was driven by her interest in exploring complex physiological models to study translocation of commensal fungi across the human intestinal barrier. And she's now a valuable member of Dynamic Food since its inception, actually, and she serves as the head of laboratory. She will be followed by Christian Maas. He's a physicist and computational biologist with over 15 years of experience in academia and industry. He received his master's in medical physics from University College of London in 2012 and his PhD from the University of Heidelberg in 2015. And he's worked as a postdoctoral researcher at the MIT in Cambridge, US until 2018. He's specialized in developing and applying digital twin for microphysiological systems and organ on chip. And currently, Christian is leading the ESQlabs division to develop further strategies integrating OEC data and computational modeling for translation of pharmacology applications. So before introducing Martin, who's also on the call and joins us for a Q&A session later, I will results. And I want to start, and I will start our second poll. In here, we are interested to learn a bit more about the applications and organ models you're interested in. So while the results are coming in, introduce Martin. He's the CEO and co-founder of Dynamic 42, and he's been working and researching with on-chip cell culture applications since 2010 already. At the Jena University Hospital, he and his co-founder Kurt Rennert first had experienced the need for physiological human organ models that properly represent the situation in vivo. So since 2021, he's also a board member of the European Network for Organ on Chip. So this is me done with the introduction, and with this, I'd like to hand over to Katja.
Thank you for the introduction and a warm welcome also from my side. Or a good morning to those from overseas, actually. I will start my presentation with a short introduction of Dynamic 42. So we are a company that offers a self-developed organ-on-chip technology. This combines not only the organ-on-chip platform, meaning the biochip itself, but also the hardware around. And we also offering education and training courses to our customers. Besides, we are also a service provider, meaning that we are carrying out in-house tests for our our customers in our own laboratories. So the company combines a lot of scientific expertise together with comprehensive biology or through decades of experience in the organ-on-chip sector. And our technology is characterized by the user-friendly ease-of-use system. So what is also very important for us is, or we are very interested in a constantly growing community. This community is not only supported by us, but we are also giving them a platform where they could exchange ideas with each other. D42 is located in central Germany. And yeah, this great work is done by the best team ever, which you can see here in this picture. Okay, so this talk will have the following outline. So we are using prednisone as a use case of high relevance in our study. But before I will come to this, I will explain a little bit what is organ-on-chip for those of you who have or who are not yet familiar with this. Afterwards, I will explain the multi-organ model that we were using in the study and will then finally come to the treatment with prednisone in this model. The computational modeling and the generation of the digital twin will then be presented in the second major part of this webinar by Christian. And finally, of course, you will have the possibility to ask questions and we hopefully find an answer for them. Okay, so let's start with the relevance of the study. So why were we using prednisone and why are we using the models I will explain later on? So by combining organ-on-chip models of the intestine, the liver, and the placenta, we aimed at finding and validating non-clinical testing methods that represent an underrepresented group, namely pregnant women. A group of people on whom it's virtually impossible to test new compounds due to ethical and moral difficulties. And especially when you think of suppression of the immune system, as is the case when you're using prednisone, even when it comes to impairment of fetal growth, as it is, for instance, shown for prednisone in animal studies, is getting more and more complicated. These non-clinical testing methods are gaining also more and more acceptance by regulatory bodies like the FDA. So there was this Modernization Act 3.0 about a year ago, so in February 2024, a new legislation was introduced by the House of Representatives. And this legislation aims to reduce and replace the use of animals in non-clinical research. And they want to improve the predictivity of non-clinical testing methods. And by a very recent press release, it was emphasized again that not only these NAMs, so new approach methodologies, but also in combination with computer modeling and artificial intelligence are needed to predict drugs behavior. So our study, let's dive a little bit into the workflow. So we aimed at determining ADME properties by using different models. We used, for instance, a gut-on-chip model to resample the uptake and permeability of prednisone. We then connected different models to each other by the vascular compartments to mimic the distribution of the drug. The liver model was used to see if the drug was metabolized. And what our main interest was, of course, the transport of the drug and its metabolite from the maternal compartment into the placenta, so in the fetal compartment. And these data from our biological experiments were then transferred to ESQlabs, and they used their computational modeling to generate a digital twin of our models that was then used to translate PK data to pregnant women by comparing them with clinical data. Okay, so I mentioned it quite a lot. What is organ-on-chip actually? So what you can read here is a definition from the European Organ Society. It says that organ-on-chip is a fit-for-purpose fabricated microfluidic device. Our chip you can see here in this little picture. And this biochip has different chambers or channels that allows the setup of living organ substructures under a controlled microenvironment. And once this tissue has developed, it can be then connected to a perfusion system that mimics the blood flow and nutrient supply similar to conditions we have in the human body. This then goes hand in hand with biochemical stimulation. So we are having a receptor activation, downstream signaling in the cells, which puts the tissue then in an even more physiological and in vivo-like state. Yeah, with this, we hope to find or we are aiming to improving the translation of data between in vitro and in vivo. So the dynamic 42 organ models are built up in a common way. And I would like to explain this by showing you our gout-on-chip model. So the system is sealed off from the outside with a top and a bottom foil, meaning everything inside is staying sterile. And we are having a porous membrane that divides this chamber into an upper and a lower channel that can be perfused individually. And this porous membrane also serves as a scaffold for cell adhesion. And by these pores, we are still having the possibility of exchanging of any kind of soluble factor. So in the gut model, we're having a vascular compartment in the upper channel and an epithelial compartment in the lower channel. And this is generated by seeding in different types of cells. So we are having human umbilical vein endothelial cells in the upper compartment forming a tight and a dense layer, and they are mixed with immune cell types. In our case, we are using macrophages that are derived from monocytes. On the underside of this membrane, we were seeding a cell line called CACO2, which is an intestinal epithelial cell line, which forms then a hanging cell layer. And after building up these models, we then connected the chip to a pump system so that both channels are perfused, meaning that the medium on top of the cells was set in motion. And by perfusing, the epithelial cells started to grow out and formed villus-like structures, what you can see here in these little bright field pictures. So the upper one showing a static chip, but once you perfuse the cells start to form a villus-like structure in a beautiful 3D architecture. This is something you only have when you perfuse the system. So for our study, as already mentioned, we are not only having the gut model, but we also used a liver model and a placenta model. They were built up in more or less the same way. So for the liver model, we also having a vascular compartment in the upper channel. They were composed of liver sinusoidal endothelial cells, also again mixed with macrophages. And in the lower channel, we seeded hepatocytes. The hepatocytes were not perfused as they are not experience shear forces in the human body. So we also didn't do so in our organ model. And the placenta model was composed of two different cell layers. The upper one in the upper channel was a trophoblast cell line called BIBO, and again using HUVEX to mimic the fetal compartment in the lower channel. Okay, so we then generated these different organ models individually in 3 different biochips, and then we connected them via the perfusion system, via a tubing system with each other. So we were building up 2 circuits. One was just perfusing the epithelial compartment of the gut model. This circulation was then later on also used for the administration of prednisone to mimic an oral uptake of the drug. And the vascular compartment of the gut model was also connected to the vascular compartment of the liver model and the maternal side of our placenta model. So this builds up then the second circuit. For analyzing our experiments, we used different readouts. During the experiments, we sampled supernatants to measure, for instance, clinical parameters like LDH. LDH is an intracellular protein that is only released in the supernatant by cell cytotoxicity. So when the cells get harmed, it is released. So this was something we were able to measure. Furthermore, we measured, of course, the concentration of our drug of prednisone and its metabolite prednisolone by analytical methods like HPLC-MS. And also we used the different compartments of our system to apply fluorescently labeled tracers so that we were able to measure the barrier functionality with the help of these tracers. And finally, at an endpoint analysis, where we recovered the tissue, so we are able to cut open this chip and to extract this membrane to fix it, and then we used it for immunofluorescence analysis. Good. To get an idea of how this looks then in reality, here are two pictures from inside the incubator. You can see here that the pump system is standing inside the incubator together with a petri dish where our chips are placed on. And on top of our chips, we are having the medium-filled reservoirs, and via the silicone tubings, they are connected to each other. As a small reminder, one of our circuits is from the gut going to the liver, then back, then to the placenta, and then back to the gut. Okay, so before I will tell you our results from our 3-organ models, we had to do some pre-qualifications or pre-tests. What you can see here is a viability measurement of the 6 different cell types we are having in all of our organ models, treated with different concentration of prednisone. So with this, we wanted to make sure this prednisone actually is not doing any harm to our cells, because our intention was to measure the distribution of the drug and its metabolite in an intact and a healthy system, and it should not have any side effects on the cells, and thereby resulting in measurements of the drug that was not intended. So luckily, we didn't see any cytotoxicity, even up to 50 micromolar of our substance. The only our positive control, Triton X100 and staurosporin, was reducing viability of the cells, which was good at this state. Then we also measured if macrophages are somehow stimulated by treatment with prednisone. We therefore measured the pro-inflammatory cytokine TNF-alpha in the culture supinatins. And here again, a concentration row of prednisone. And what you can see here in comparison to our control cells that were non-treated cells, we don't have any higher levels in TNF-alpha. This is simply just background levels here. You can see our positive control LPS, which is a bacterial antitoxin, is releasing much more TNF-alpha from the cells. So we also have no effect on the immune cells that are present in our system. So another pretest we had to do is to measure the adsorption of the drug to the chip system. This is something we always recommend to do so at the beginning of these experiments, because even though we are using a biocompatible material for our chips that is also characterized by low adsorption properties, still the plastic surface is much higher than in a normal well plate. And we therefore used a chip system that was not seeded with any cells and applied 3 different concentrations of prednisone and measured it after 24 hours, the remaining concentration. And also luckily for us, we didn't see any adsorption to the plastic material. So the concentration we intended to have, or we wanted to have in the system, is really also then present when we're having cell-seeded chips. Good. Then let's get to the organ-on-chip models. This 3-organ model was, or this was the first time that we connected these 3 organs to each other. And therefore we needed to make sure that they're really forming a robust connection to each other, that are not influencing each other. And we therefore measured the cytotoxicity by the release of LTH in the supinatins after connecting these 3 organ models. So what you can see here are LDH values from all the 6 different compartments of the 3 different models. Always comparison single chips to the connected setup. And what you can see here that even though we have variations, which is just normal in our complex models, we are not having higher cytotoxicity upon connection of the 3 models. What we also measured was the permeability of the barrier of our 3 models by introducing FITC-dextran, as I said, a fluorescently labeled tracer. If this goes through the barrier, we are having, or by this we can measure actually the permeability. And the higher the permeability for phytidextrin is, the lower the barrier integrity is. But yeah, luckily for us, we also kept compared here again single models to connected models, and the barrier was always the same or even better. So lower permeability values here for the placenta models, meaning a very good barrier integrity. Good. So with these results, we were able to start. We had a last quick view on cell integrity by doing immunofluorescence staining for different marker proteins of our different models. Just for instance, shown here the CACO2 layer forming our villus-like structures. This was the case no matter if the chips were single-lead, perfused, or in connection to each other. Good. So then we started with the prednisone treatment. We applied the drug in the epithelial circuit or circulation of the gut model, and then let this incubate or perfuse for 24 hours. We again checked that prednisone itself has no effect on the cells, so that it's not inducing any cytotoxicity and leading to false value of distribution of the drug between the different compartments. This is shown here in this diagram. So again, measuring LDH and comparing this to single models that were non-treated. And what you can see here, no matter if we are connecting the models to each other or if prednisone is also present in single models and connected models, the value levels stay the same after 24 hours of prednisone treatment. I forgot to mention that we are using always the plasma concentration or the Cmax of prednisone, which was 55 nanomolar. Then again, our permeability assay, now also not only connection versus single, but also after treatment with prednisone for 24 hours, you can see here the values of permeability are rather decreasing, which is good, meaning we're having a tight and a good barrier function of the three different models. Oh, this is actually my last slide, showing you the concentrations of prednisone and prednisolone in the 6 different compartments of our 3-organ models. We took samples after different time points of the 10, 20, and 24 hours and measured via HPLC, MS, the concentrations of the 2 compounds. This table here summarizes the results, and I will quickly guide you through. So we are applying the drug on the epithelial side of the gut model. We therefore see here quite high values that are staying more or less the same over time. This prednisone levels, or a part of this prednisone, you can also then find in the vasculature, meaning that it's transported from the epithelial site into the vascular compartment. And why, or because the 3 models are connected to each other, you see that you can find prednisone also in the vascular compartments of all other models. It's increasing over time, which is good. And what we were, of course, also interested whether prednisolone can be found. And what you can see here is that prednisolone is generated, but only in the epithelial compartment of the liver, meaning by the hepatocytes. And this is also increasing over time. And then this prednisolone is also transported in the vasculature. That's the reason why you can find it in the 3 vascular compartments of the 3 organs. And what is most important for us is the result over here. Even though we can have prednisolone in the maternal part of the placenta model, we don't find it in the fetal part, meaning there are some exclusion, or that there is exclusion happening, even though it is present in the vasculature of the maternal part. Okay, so these biological data would then transfer to Christian, who used them together with his computational modeling to generate the digital twins. And with this, I will hand over to Christian.
Yeah, so thanks again also for introducing me and welcoming me to this great webinar. I'm really thrilled to dive in the next 20-ish minutes or so into more the computational part of the work, and basically sort of like, highlight what we did and how we get actually from chip to clinic. A bit of background sort of like on the company. So ESQlabs, as was nicely introduced by Juliane, is a business unit of ESQlabs. So we are a a German startup, we do biosimulation basically. We have 3 clear goals, right? Patient PK and safety. So this is one that we will be touching up on in this presentation. 3R, and of course, saving animals. Really, Katja mentioned it, the push with the FDA recently again is really very timely. EMA finally has to step up, and something that I wish we would have had 2 years ago when we did the study, but reducing sampling time and cost and effort, of course, which might be interesting for some of the folks here on the call. Quick overview of the company. We are right now 3 people with some expertise from a colleague that's actually half employed by us and by ESQlabs, and of course the mentorship and guidance with Stefan, CEO of ESQlabs. Our partners, just to like quickly highlight and sort of like show you the breadth and the width of the work we do, and also not to forget the not to be forgotten the regulatory engagement. Very important to work on the standardization roadmap. The 3 pillars that we basically work is in operational design. So we really want to focus on helping most of the guys, I guess, on this call, replicating human PK in vitro. I mean, nowadays everyone understands that exposing cells to sort of like a constant concentration is more and more sort of like updated with new tech where you can actually replicate this. Then I will be diving into the digital twin integration, of course, first on the organ chip and MPS side, but then secondly also on the human PPK side to sort of like simulate then eventually the kinetics in not only a pregnant mother, but then of course in the fetus. And then that is sort of like outside the scope for this one. Of course, we also work in AI and machine learning, but granted, in the biotech space, there's not a lot of data. So it's really difficult to get a good data set where you actually can train a model. Now, one thing that's really close to my heart, and then I will dive into the work that we did here, is the virtual testing center. That's sort of like our virtual hub that basically offers, interpretation, analysis, and integration and translation of chip results, and sort of like end-to-end analysis to the human situation with different, contexts of use and application scenarios. And I will be presenting some of those in this case. One more really important aspect here is benchmarking. So if you know, whether you are a pharma employee or like, you are just doing your PhD at academia, it doesn't matter. If you type in sort of like, what's the best liver chip, you will get a different number of companies in your favorite online search tool. And the question, of course, is, okay, what's the best chip for my particular context of use, right? Or like the question that I want to study. Of course, the guys from Dynamic 42 are doing a great job, but there might be other companies out there who, with a different context, actually could perform slightly better. And we have a tool to sort of like offer benchmarking this here, in this case, for clearance prediction of human situation based on liver chip experimentations. Okay, now sort of like taking back a gear and diving into digital twin. So I think there was a bit of an outline. So that's it's a very busy and complex slide, but that's sort of like the setup that we actually did within this project. So while we, of course, put ourselves here at the center, it's the interlinking nature of this workflow that we have the bio guys producing the data, but of course, we inform some of the experimental design. Then out of these experiments, you actually get data, but not only drug PK and PD, but also biomarkers, right? To sort of like relate the exposure and the readouts that you see and be able to interpret this. On the other hand, of course, we had the sponsor that provided the drug and the question of interest in this case. And then, of course, at the end, ultimately, what we always want to do is we want to predict how the situation is actually in a human being to then eventually either guide earlier and better decision-making processes, but eventually, of course, to fully replace animal studies that we really actually don't need anymore. Now, Katja showed this, we saw this, they run the single NPS studies, right? And what we actually got at the end was a big Excel sheet with some of the concentration values. And this is actually still in German here, and some concentration and time values where this was measured. And this is cool, this is great, right? But it doesn't tell you anything. So what you of course can do is you can start to plot the data as a time concentration profile. But even then, the question is there anything more in it? Can you actually get anything more out of just plotting your PK data? Because from that, you did a cool experiment and you see that there's a change in your drug concentration, but it's, it requires some analysis and some interpretation to really understand what's going on. Yeah, so the digital twin approach that we, or when we talk about the digital twin approach, we basically mean what I want to present here on this slide. It's a term used over the past 5 years at very high speed. You read this everywhere basically, so it requires a bit of a definition. And we really think about this as a computational model that tries to emulate the biological processes that you guys, or most of the bio guys, I guess, try to emulate on the lab bench. So we're not only interested in the actual biological processes, right? For example, liver clearance, but also drug permeability. So it's very important to understand how and why a drug actually is permeating and partitioning from the cell culture media into the interstitial, and then of course into the intracellular part. But of course, I mean, we just saw it on the benchmarking slide. There's a difference in chip architecture, so media volume, cell number. Sometimes these chips have flow, some have flow on the apical and basal side if you connect multiple chips together. So that's all information that you gotta take into account to also pay tribute to the fact that there are ever more complex biomodels being developed. So you have to analyze them with more complex math models to account for the fact. And then, of course, if you make the math models more bigger, you have more parameters, and you have to fill in these parameters with values. Otherwise, what's the sense? And we do this with so sort of like drug specific. So we know all of the physchem properties of these compounds that we are studying, and there's retrospective and prospective, right? So we take log the lipophilicity in the media binding, for example, and we put that into pre-existing Q-cell models that then help us to really preemptively calculate some of these parameters and thereby sort of like disentangle biological processes from each other. How does it actually look like in our setting? So here for the liver, right? So on the left-hand side, that's a nice graphics from Dynamic showing, excuse me, how their system basically looks like, right? So that you see the flow, you see the cells, the membrane. This looks perfect. And to me, as a physicist, I'm always amazed on, what you actually can do with these tiny chips and how cells can survive in there. The language that I'm speaking is a bit more here in the middle, right? Hence the lower graphics. So we actually put all of this nice graphics on the left-hand side into boxes, into compartments. And these compartments basically represent a physical space on this physical chip, if you want. And the arrows basically tell you how a compound actually gets from A to B. And in this case, this is linked to permeability and partitioning. These are the governing basic physiological processes that happen basically in any cell, for that matter. And then in that case here for the liver, we actually see that the clearance now, occurs at the intracellular part. So my bio knowledge is limited, I would say, but I learned at least that the enzymes are not, in the media, but they're actually sort of like, some are at the cell surface, but some are actually intracellular. Expressed, so the drug has to get there in order to be metabolized. That's the main idea. Now, so what you then can do if you apply this mathematical model to the data that I just showed, you can start to put a line through these dots, right? And the line basically can be converted into a on-chip liver clearance value, which you need another mathematical model to then translate to humans, right? But in this case, we actually wanted to see whether the mini tiny bit of the human liver that we emulated on the lab bench basically represents quantitatively the human clearance value of the same compound, and it did, right? So that's really nice. That's a good check mark to like really understand how you can use this data, but also how to interpret this and to analyze this. Back then, we just did this, with our coding. Now we have a nice GUI actually behind this where you can select from different compounds and different chips, basically, and then you can sort of like, so for those people interested in not coding themselves, but actually having a code or like a software do this in the back end, there's now also like a nice GUI behind us that makes it a lot easier. Now, for the gut, the same thing. And if you look carefully, right, the graphical representation here with these boxes now looks a bit different. And reason being, as Katja mentioned in the beginning, the top and bottom channel now have a connection to a reservoir where also a systemic recirculation and flow basically is being applied. And that's just basically due to physiology and biology, right? But we need to understand that this is the case because it basically tells you how and where the drug basically is moving within the system and how can potentially get from the bottom channel to the top channel and vice versa. Now, by looking at the data here, right, what we wanted to see is that there's a hugely favored influx or also, uptake. So we wanted to know that if you orally dose the compound, whether the lab system can basically mimic a human comparable situation in terms of uptake, and that was nicely the case. So that's another check mark on this biological function. Now, last but not least, of course, the placenta. Here we wanted to see a similar thing. And again, if you look very, very carefully, the graphical representation here now lacks this bottom reservoir. And that again is due to the hardware and the fact that the placental, that the vivo cells on the bottom channel basically don't like the flow, according to biologists, right? So we didn't add this in the experiment, and then of course also we had to remove this from the computational model to describe correctly how the drug is flowing within the system. If you then analyze the same permeability PK data in the setting, what you then basically get is that you now have a hugely favored efflux. So that means there's an active pump, a transport pushing the prednisone from the placental side to the maternal side, right? And that's exactly what we observe also in the clinic with the very limited data information that is there. Now, one thing, and Katja showed this, is then, of course, we wanted to predict this. How would this actually look like in a three-way interactome? And this, again, picture that is very, very super wow for me that you can actually do this. I mean, I would lose sort of like control over what tubing actually means what. Again, my language is more this graphical representation here in the middle, and we expanded sort of like a digital twin approach to match this more complex bio part, right? And then the idea was that we want to predict, first of all, the on-chip PK before we actually run an actual bio experimentation. And we did this by implementing the data from the single NPS systems in this computational twin, and then simulated this. And then we overlaid the experimental data. So technically, that's a true validation here. As you can see, so I mean, sure, the red curve, is like, you could potentially map this a bit better. Here's a bit of an outlier, right? But overall, what we can show is that the single NPS and the digital twin actually qualifies and validates the three-way interactome. Now, a very bold statement, and I don't want to put Dynamic 42 here out of business, right, is eventually that actually tells people that you don't need to run these complex three-way interactome studies for that particular context of use, I have to admit, right? The semi-bold statement, of course, and that's definitely the way for the next 5 to 10 years, is that you, rather than running 5 or 10 very complex 3 or 4, 5-way interactome experiments, right, where there's a lot of failure rate, of course, because the systems are getting more complex, you could run maybe only 1 or 2 experiments and then complement some of these experiments with computational simulations, right? So that's the outcome of this study, and that's a very nice one because it's obviously very timely, cost efficient, and saves a lot of lab work. Now, in order to also do this, and again, I wish we had that this tool also back when we did the study, because I kept telling the dynamic guys, no, you have to measure here and here, like we need all of this data. But with our new approach that we call MICT, you actually have a sustainable and faster, accurate experimental study design where we can save up to 60% of the samplings. In this particular study, we could have actually reduced the samplings from 48 samples. So we measured from the gut and the placenta and the liver and like the top channel and the bottom channel. So like there was a lot of data, and it was quite cumbersome. I guess Katja will agree with me later. But actually, sort of like with the computational model behind this, we could have reduce this to by about 50%. So, and that is, of course, a lot of cost savings and manual work that could be saved down the road. Now, but sort of like switching gears back, now that we did run these simulations for the single MPS systems and the integrated chip systems, now we wanted actually to see, okay, how does it look like in a human being? For this, we first need to develop a computational model of prednisone in a pregnant woman, right? So yeah, we implemented this PPK model. And then sort of like use the information from these individual chips and sort of like plug and play like a Lego Frankenstein type of monster into this human computational twin, and then we sort of like hit the button and simulated this. And as you can see here, again, it's a very busy slide, but at the end of the day, the black dots is PK, so actual plasma concentration in pregnant women out of a clinical study after a single dose of prednisone. If you take the literature values that you find and like you try to simulate this, you actually get this green curve. So like, there's a bit of a mismatch between the information that is being published and how pregnant women actually respond to prednisone. If you take so if you fit some of the values, then you actually get this red curve, which is very, very nicely alongside the data, but more importantly so is if you plug in our single MPS data, you actually get this blue curve. And granted, while we are slightly under-predicting this PK curve, it sort of like follows the same trend. And that's really interesting and really nice to see, because we had a comparable liver clearance already with the chip, and we saw qualitatively that the gut favored uptake, while the placenta showed that there is efflux happening. Now, again, that was still not the final goal, right? So the final goal was actually to put all of these pieces together and then predict this at the end in the fetus. So here we simulated this over 3 months in the maternal, so simulating that a pregnant woman would take this prednisone pill at 10 milligram every day. And as expected, there's sort of like no accumulation, right, in the maternal side. But very interestingly, we simulated an accumulation effect on the fetal side. So that was quite interesting to know, even though we saw that there is huge efflux actually from the placenta side. So that has to do with the partitioning and the maternal plasma to fetal plasma ratio. But long story short, we actually sort of like had a steady state concentration of about 50 nanomolar, which is considered, within the literature as being very, very safe. And this is also something that we tested on the liver gut, but also more importantly than so on the placenta cells, right? So we would agree here that there's a very, very limited risk for an unborn baby. Now, to sort of like wrap this up and put this all together, so, we can match the clinical observation with the MPS-based results, but also more importantly, so after the analysis with digital twins, we can predict the human PK, and that, of course, would help early go, no-go decision, and very nicely without the use of animals, right? Granted, it's a showcase study, so we would need to dive into this a bit deeper and test this on more compounds. But there's a lot of evidence here. And of course, very, very interesting is to sort of like be able to reduce very complex, these multi-organ studies down the road with a combination with organochips and digital twins. Lastly, I would like to thank the team, our sponsors and Dynamic 42, as well as University Clinic Jena, Diana and Jose. And with that, I would like to thank you for your attention.
Thank you. Thank you, Christian and Katja, for your presentations. I think they provided a really good overview of our joint study. Before I move into a Q&A session, I'd just swiftly like to launch our last survey, because we would like to hear from you how you liked the webinar so far. Okay, so let's have a look which questions we got so far. Okay, so the first one is actually directly back to Christian. How did you validate the three-way interaction predictions of the fetal exposure to prednisone, and how does it compare to the actual clinical data? You're on mute, Christian.
Sorry, wrong button.
That's all right.
So the actual validation of the three-way interactome from the lab was what I was basically showing, right? So we had the single NPS information, liver clearance, gut permeability, and placenta permeability, and we plugged those data into the three-way interactome digital twin. We pushed a button, simulated the kinetics, right? And then we had the kinetics of the three-way interactome. And then we overlaid this with the actual data that the dynamic 42 guys measured. And that matched pretty closely. So that's a direct validation sort of like within this setting. Now for the second part of the question, the translation to humans and validation is the first step was the qualification, I would say, with the model to be able to describe maternal plasma PK. That's the best you can do, right? There is no more data, basically no more data out there. And then the only data that actually is available is the ratio of maternal and fetal plasma of some of the compounds. And you can turn this into a partition coefficient, right? So like this basically tells you how much drug is going from A to B, whereas permeability tells you how fast the drug is going from A to B. And we matched sort of like so we took the placenta permeability, that's sort of like the rate, but the extent of this was then matched to this ratio that we saw from clinical studies. So that's sort of like that's an indication. And then secondly, I would be bold enough to sort of like say the concentration level that we simulated based on the NPS results is way below, known clinical tox concentration. So you are on the safe side anyways.
Thank you, Christian. Actually, the next one's also to you. Do you believe that patient diseases could be plugged into a computational model to predict changes in PK?
Can you repeat?
Yeah, sure. Do you believe that patient diseases could be plugged into a computational model to predict changes in PK?
No, perfect. This is sorry. We just had a discussion this morning, so hence my smile. So stick to the example of NASH or NAFLD, right? So like there are some liver chip companies and publications out there where you have a healthy liver chip and a diseased sort of like state. In the bio lab, you can then basically you can look at your biomarker, at your gene RNA sequencing and whatnot, and you can basically say, okay, I have a healthy liver and I have a NASH liver. Now, the idea is that you then sort of like take these biomarker differences and RNA sequencing differences and sort of like extract, for example, differences in CYP3A4 enzymes, right? Or any other metabolizing enzymes for that matter. That information can then be an input into a human digital twin, into a chip digital twin, where you then can simulate different kinetics, right? And then you see sort of like what the effect is on your chip already, healthy versus disease, right? And that information then again can be taken into the human digital twin, where you then can say, okay, what if you have a healthy person with a certain CYP enzyme expression that shows a certain PK kinetics of the drug, and that is then linked to efficacy, right? So, or say you have a tumor patient, right, but has a healthy liver. If you have a NASH liver and also like the same tumor, right, then you will definitely see a different PK, and that has implications for your treatment efficacy down the road. So yeah, I hope this sort of like makes sense a bit. There are definitely ways to implement this, and you can start as early as multi-omics RNA sequencing from the chip to then understand this and then put this into your human models.
Yeah, makes a lot of sense. Thank you. So the next one is to Katja. How is perfusion through 3 chambers with different cells possible? What are the requirements for the medium?
Yeah, for sure. Of course, there are requirements, and different organ models have different kind of media results, and different regions that have different, let's say, expectations of what they need. And we were lucky with our models. So the 3 different organ models we connected to each other actually use the same medium in the upper channels. So for the vascular compartment of the gut and the liver, we use the same medium, but also for the vivo side, for the trophoblast of the placenta model, we use the same medium. So there we didn't have any problems by connecting them because we could use the same medium for all of them. But yeah, transferring this to maybe other models that can be connected to each other. You can think of lung liver models, for instance. There you need to do pre-experiments on this. So if they have different media, you need to test if they feel very well with one or the other, or if you have to do a mixture of the medium, and then of course do pre-test on viability of the cells, on a barrier function and so on, if you see that it's still the same no matter which medium you are using. Okay, I hope this answers the question.
Thank you, Katja. And next one, I believe, is to Christian again. You mentioned to reduce experimental costs by using MIQED. What is MIQED?
Yeah. So because the acronym is so difficult to pronounce, I thought to be cool and want to call this MIQED, although they're like it has nothing to do with music. So it stands for Model-Informed Quantitative Experimental Design. So it's a long buzzword, right? But what this does at the end is here. So what this does is it takes and it works best for multi-organ chip systems, right? So in this case, we have the gut and liver system, and we wanted to see, or say, it's a mathematical approach from information theory, where you basically want to understand, did I measure at the correct time points, or is there potentially a way to measure at different time points, maybe more often or maybe even less, ideally, right? Such that I get almost as much information as I got with my typical sampling schedule. And we run this here for a gut liver study where we wanted to look at, we wanted to estimate clearance, and you see like the PK from the apical side and basolateral side, and then we sample from the mixing chamber. And the green dots is your typical profile, right? So you measure at time 0, at 2 hours, 4 hours, 6 hours, I don't know, 18 hours. So hopefully no one went into the lab at night, right? 24 hours, and then 48 hours, 72 hours. So that's a lot of data points for like a simple PK study, right? And we always get this question, like, is there anything you can do to reduce this? Well, it's here on screen. We can. So the red dots is basically what we have as the optimized sampling schedule, and it's quite a few data points only, so it's about half. And as you can see here, the root squared error basically is 25%. That means your estimation of liver clearance is 25% more uncertain than it would have been if you would have measured all of the 8 or 9 time points before, right? So yeah, it's up to you, I guess, to decide, what uncertainty you can basically live with. But there is a way to sort of like make this like a very quick shot. And then, of course, if you have, if you can get rid of a few of those last data point, this would be super cool, right? Because then your experiment only runs for 48 hours and not 72. So you have less experimental time, less labor, and of course, like you have a lot of, lower cost per sampling. And again, I didn't show the graphics for the gut liver placenta because it was like 9 compartments, so like 9 times PK curves. But again, the outcome is basically the same. We could have measured only half of the data points. I hope this answers the question.
Okay, so the next one is for Katja again. What adjustments were made to the concentration of prednisone when comparing single versus triple model?
Yeah, for sure. We had to make some adjustments there. I didn't went into detail that much just because I didn't want to make it too complicated during my talk. But we, of course, having different volumes of medium in a connected model compared to a single model. And we aimed at having a DC max concentration of 55 nanomolar in the circulation. That's why we had to increase the concentration of prednisone in the gut epithelial compartment. We raised it up to 140 nanomolar. But when we are thinking about a full transport or influx of the drug in the vascularization, then and dilution of the drug, we ended up at 55 nanomolar, at least due to our calculations. And by what we actually saw by our analytical measurements, by LCMS measurements, is that we were right with our concentration. So we ended up around 50 nanomolar in the vascular compartment. So yeah, therefore we had to do some tricks at the beginning.
Okay, so speaking of tricks, I guess, another question is, how are the three models synchronized? So what are the different cell types of a model? How do you seed them at the same time? Yeah, how does the seeding work, basically?
Yeah, this is really depending on the model. So they all have different requirements when building them up. So the blood-placenta model, for instance, is very easy. You can seed in both cell types at the same day, one in the morning, adhering to the surface, and one in the afternoon. So basically the next day you are ready to go for perfusion. But with the liver and the gut model, it's a bit different. Liver model also takes up to 3 to 4 days. So they're seeding in one cell type after the other. So one cell type per day, meaning that you need about a week until you start perfusion. And with the gut model, it's even a bit longer because we wanted to have these differentiated and cells of our epithelial cells, so that they were forming in villus-like structures on the flow, and this needs some time. Even though it's much quicker than a well plate, the cells are usually differentiated for up to 3 weeks. We already see these villus formation within a few days, but still you need to start earlier with building up the gut model than these 2 other models. This is something you need to find out at the beginning and then synchronize the timelines of the 3 organs.
Perfect. So, and well, we're approaching the end of the webinar, and there is actually only just one question left. If you do have one more, submit it now. I might actually be able to take it, but otherwise this will be the last question. That's going to Christian. How scalable is your approach for evaluating other drugs in pregnancy, like especially ones that have limited human data?
Fully, completely, boom. So it works. Like, so we know, so retrospectively we have a lot of first-scale information, right? So log P, molecular weight, solubility. So that's basically all out there. We have a high throughput PPK model approach as well, where you basically take all of that info, ideally some in vitro data if you have it, or some animal data, just to make use of it, not to run additional animal studies. And then you can, like simulate this and compare it to as many, data points that you can basically find for those drugs. We did the biggest study with liver chip clearance, right? So we have 80 compounds on this and data from published sources to do that. But yeah, ideally we have, some in vitro measurements with a placenta model to understand how compounds go from A to B. And you take that knowledge, really plug it in, and then sort of like confirm whether your PK is exactly as you imagine it to be, and if not, that's even better because then you can learn something and understand, can make the model, the biomodel, and the computational model more complex and advanced, I guess.
Right. Well, that's a nice ending. So yeah, with this, we're at the end of our webinar. Thank you everyone for joining us today. And as I said, I will send out the recording very soon. So goodbye.
All right. Thanks a lot. Goodbye.